Hello students, today we'll start with the new topic drug discovery. A drug is a substance, chemical molecule that is used for treatment, diagnosis, prevention or cure of a disease or intended to affect the structure or function of a body. Based on its medical use, drugs can be classified as pharmacodynamic agents, central nervous system agents, chemotherapeutic agents, and supplement agents. Pharmacodynamic agents are drugs that affect the physiological function of the body. Example, analgesic, anti-allergic, anti-inflammatory drugs, cardiovascular agents, etc. Central nervous system agents affect central nervous system. Example, antidepressant drugs, anesthetic, etc. Chemotherapeutic agents are drugs that help to fight pathogens. Example, antibiotics, antiviral, antifungal agents. Supplement agents are vitamins or dietary supplements. Drug design is an inventory process to prepare drugs based on knowledge of biological targets. A drug could be a small molecule or ligand that mimics the biological molecule and brings about therapeutic response. There are many approaches for drug designing. Presently, drug designing depends on computer-aided or structural-based drug designing. Drug design can be broadly divided into two phases, drug discovery and drug development. In drug discovery, finding the lead component or lead molecule is a key step. The lead reactive component could be obtained by various approaches. First, screening of biologically active compounds from plant sources or already known bioactive molecules. Second, alteration of the chemical structure of an existing active molecule. Third, discovery of a novel molecule, or by identification of new targets for the molecule. Drug development is the characterization of ADME properties, toxicology, and dosage studies, followed by marketing strategies for the drug. The steps in drug discovery include first, identification or choice of disease. Second, drug target for the disease. Identify a bioassay for it. Find a lead compound. Isolate the lead compound and determine the structure of the lead compound. Identify structure-activity relationships. Identify the pharmacophore and improve target interactions. Steps or strategies for drug development are first, improve pharmacokinetic property of the lead compound or the modified lead compound, toxicological evaluation, designing a manufacturing process, carry out clinical trials, market the drug, and post-market monitoring and inspections. The figure here represents the drug discovery and development timeline. The current drug approval pipeline can take approximately 15 years. It is estimated that around 5,000 to 10,000 compounds are selected and only one of them reaches the drug market as drug. Drug discovery methods are of two types, traditional drug discovery or forward pharmacology and rational drug design or reverse pharmacology. Traditional drug discovery method is the conventional method of trial and error based. The presumed bioactive molecule is tested in vitro systems followed by in vivo methods and then used in humans. The process direction is from lab to clinic. Rational drug design is a systematic approach of drug designing. In this method, the target is identified and the molecule is designed and tested using computational approaches, structural based designs, QSR, etc. And drug likeness of a compound is usually predicted using Lipinski rule of 5 or lipophilic efficiency. Traditional drug discovery. This method is a trial-error method. Compounds are screened for bioactive molecule. In vitro bioassays are conducted to check the bioactivity and characterization of the molecule is done. In vivo tests are also conducted. After a battery of tests, the lead compound is identified. The activity of the lead compound is improved or made more effective by modifications followed by pharmacokinetics and toxicology studies before going for clinical studies and FDA approval. This is followed by marketing strategies. The flowchart below illustrates a conventional drug discovery approach. Methods of traditional drug design. It includes random screening, trial and error method, ethnopharmacological approach, serendipity method, and classical pharmacology. The random screening approach involves randomly screening natural or synthetic compounds for its bioactive properties using certain bioassays. It could be based on choosing compounds based on structure, example alkaloids, terpenoids, phenolics, or could be based on screening of plants using certain selected bioassay. The trial and error method. As the term suggests, different parts of the plants are used to give the desired response. Next, ethnopharmacological approach. It involves study of indigenous drugs and then going for experimental investigation. Many of the present day drugs that we have are formed or found from this approach. Serendipity method. It means accidental discovery. Example, penicillin as an antibiotic was accidentally discovered by Alexander Fleming. Classical pharmacology. In this method, the drug is identified based upon function. No prior identification of the drug target is there. Example, vincristine from periwinkle plant. Here, the method is lab to clinic. That is why it is known as forward pharmacology. The main disadvantage of the traditional drug discovery method is it is expensive, time consuming, and often ineffective or inefficient. The conventional drug discovery process was a result of unsystematic, random, and serendipitous approach. The rational drug designing method is a methodological approach. It is a transdisciplinary approach of integrating traditional knowledge, experimental observation, and clinical experience. Here, development of the drug is based upon hypothesis. It involves the identification of the receptor or the enzyme that is relevant to a disease, elucidate the structure and function of the receptor or enzyme, and then design a drug molecule that will possibly interact with the receptor and enzyme to give a therapeutic effect. The diagram here represents a rational drug discovery approach. We'll study about the different methods used in rational drug discovery design in our future sessions. In this method, the discovery process is from clinic to lab. Hence, it is known as reverse pharmacology. The figure here illustrates the differences or comparison between the two approaches of drug designing, the classical or conventional method and the rational or the reverse pharmacological method. Thank you.